So one of the most commonly used methods for determining nitrogen content is a method called the Kjeldahl method. And it was developed in 1883 by a Danish scientist named Kjeldahl, so it was named after him. And even though it was developed in 1883, it's still really widely used today, as is one of the most reliable methods for determining nitrogen in food, in water, or in any sample really. So it's a three-step process that we're going to go through. It starts with the digestion. So right here we have a digestion block, and this heats up the sample to a really high temperature and digests the sample with sulfuric acid and a catalyst to break down the organic nitrogen into ammonia, and the ammonia reacts with the sulfuric acid and produces ammonium sulfate. So once that's done, that runs for about a half an hour or so, then we let the tubes cool down. This is a scrubber which cleans up any of the harmful vapors that are given off. I'll explain that in more detail later. So once the digestion is done, then we move over and we do a distillation. So we have another piece of equipment for that. So in the distillation, we add a strong base to it, so sodium hydroxide in this case, and it is a steam distillation that distills the ammonia from the sample into boric acid. So we put our sample in here. Inside this, the pieces you can't see, is a heating coil and a condenser, so the ammonia comes out into our receiving flask. And then the third and final step would be a titration, and we're going to use a digital titrator with hydrochloric acid to determine how much ammonia is in the sample. So we're going to start with our samples. So this is the digestion tube that we use to put our sample in, that we add the sulfuric acid and the catalyst to. It's basically just a large test tube. So we're going to start by measuring out our sample into the tube. Since we're doing a protein lab, for our samples we're going to use meat products. So we have here on the left a sliced ham product, and on the right we have the same product but it's been homogenized. So I've just ground it up so that we have a homogeneous sample to use for our nitrogen determination. So now we're going to take a small amount, between 100 and 300 milligrams of the homogenized ham sample, into our digestion tubes. So now we're ready to start digesting our samples. So we have in the tube about 300 milligrams of the sample, of the ham sample in this case. And I also have in there a Kjeldahl tablet, which is a catalyst tablet. So if you look closely at that, you'll see a little tablet in there. And that just helps the reaction proceed. And then to that, I'm going to add 20 milliliters of sulfuric acid. So this is concentrated sulfuric acid, which is why I'm wearing gloves, and doing this in a fume hood. Anytime you're dealing with corrosive acids, you want to be very careful. I'm pouring it down the side of the tube. Okay, so that's ready to go. Now we will take them over to the digestion block and get that running. Okay, so now we're over here at our digestion block. So this is the digester, and you can see that our tubes with our samples in it have been placed into the heating block. So in here, underneath the samples, what the samples are sitting in, that's our heating mantle, and that will heat up to about 421 degrees Celsius, so very high temperature. On top of that, you'll notice this piece of apparatus, that's called a scrubber. And what that does is it takes care of any of the harmful fumes or vapors that are being given off when it starts to heat up. So remember, we put concentrated sulfuric acid in these tubes. So when the sulfuric acid heats up, it's going to give off a lot of vapors, and we don't want to be breathing that in. So the scrubber takes care of those vapors and neutralizes them before it releases it into the fume hood. So this is the part of the process where the proteins or the nitrogen in the sample is broken down into ammonium sulfate. Okay, so the protein in the presence of sulfuric acid under heat and with the catalyst produces ammonium sulfate. And then when that's finished, we will go on to do the next step, the neutralization and distillation. So we're going to let this heat up. You can see that it's only just starting to heat up now, so we put it on when it's cool and we let it heat up gradually. and then we'll let it run. Once it gets to 420 degrees, we will let it run for probably 20-30 minutes until the sample is completely digested. And you'll be able to see when it's digested because we'll see a color change in our test tubes. So see that when we come back. So our digestion block has been running now for about 30 minutes, and you can really see now those vapors that I mentioned. You can see that the samples are giving out a lot of vapors. You can't really see the samples, you can see them a little bit. You'll see those better when I can take them out. So the scrubber is working now. Scroll over, you can see it's bubbling away. So this works in that it has two solutions that the vapors are bubbled through. So we have a sodium hydroxide and then a water. And what that does is it neutralizes those acidic vapors that are being given off by the samples in the digestion block. So that's pretty much ready there now. So I'm just going to turn it off. We're going to let it cool down, and we'll be back shortly. So our samples have sufficiently cooled down. They're all digested. So now what we are going to do is add 75 milliliters of water to each one, just to dilute it before we do the distillation. So I'm just adding water, and I'm doing it carefully because we're adding water to acid. So we tend to get some vapors given off when we do that, which you can see there. So we're still working in the fume hood as well. So we add 75 milliliters of water. And now you can really see that bluish green color. So that's the color we would expect to see. So now that those are all done, we're going to go ahead and start our distillation. So now that our samples are digested and cooled down, we're ready to start the distillation. So this is an automatic distillation unit. And inside this machine, what you can't see, there is a heating coil which heats water to produce steam, and a condenser as well. So we heat the sample, which liberates the ammonia, and then it is distilled by steam for about 5 to 7 minutes through a condenser, and then the ammonia is collected in the receiving flask. So when we put our sample on, so our sample tube fits in here. And this tube that you see in the middle is the tube that supplies the steam. So make sure that's in there nice and secure. It has a safety door so that if there's any splashing of alkali or anything, it stays contained within the apparatus. This is our receiving flask, and in this flask is 25 milliliters of boric acid, and it also contains an indicator. So there's an indicator in there that will change color when the end point of our reaction is reached. So this is an automatic titrator. Once we have our sample and our receiving flask in, we can press start. And you should see fairly soon here now the alkali being dispensed into the tube. So the alkali in this case is sodium hydroxide. There you go. So that's dispensed in the tube. And you can see that that's a fairly vigorous reaction because it's a concentrated base reacting with a concentrated acid. Remember the sulfuric acid that's in our tube. And once all the alkali is in, the steam distillation starts. So you can see the bubbles being produced there in the tube. So that's being pumped into the sample tube through the steam tube. And that will run now for about 5 to 7 minutes until all of our ammonia is collected in the receiving flask. Okay, so our distillation is complete, and you can see that this machine automatically drains our tube and dispenses that into waste. Our receiving flask you can take out, and you can see now that it has changed color. So it's a greenish color as opposed to the red we had when we started. So that means that our ammonia has been condensed into the receiving flask, and now we're going to perform a titration with this solution. So just so you can see the difference in the color, the flask on the right is the original color of the boric acid with the indicator before the titration, and now we can see we have this greenish blue color of the solution after the titration. And the color change is due to the ammonia that was liberated from the sample reacts with the boric acid to produce ammonium borate, and that's what produces the color change. So now we're going to titrate this with a solution of hydrochloric acid. So we're using a digital titrator, okay, rather than a burette. And we're going to titrate that with hydrochloric acid, and the volume of the titrant that we use is the value that we'll use in our calculations to determine the amount of nitrogen in the sample. Okay, so let's start the titration. So I'm just adding hydrochloric acid very slowly at first, and we're looking for a color change from the green color to the original red color of the boric acid with the indicator. Okay, there we go. So you can see we're right back to that original color that we started with of the boric acid. So we've reached the end point of our titration. So the volume that we added will be the volume that we'll use in our calculations that we can use to calculate the amount of ammonia in our sample. So I'm going to run 2 more samples so we have triplicate results, as well as a blank, and that's the Kjeldahl nitrogen method.